Hello everybody, welcome to episode 2 of Intellectual Interlude, your podcast brought to you by the British Science Association Derbyshire branch. I was going to say your weekly podcast, but there wasn't one last week, so we're keeping our options open in terms of the regularity. We're hoping for it to be regular, but obviously last week things got in the way, but we're back this week for episode 2. I'm David, treasurer of the branch, and with me as usual is Ian.
Yay! I am the secretary of the branch, and we have a special guest this week.
Hello! My name is Agatha, and I'm a molecular biologist, and I'm also involved in British Science Association. I used to be a secretary.
I got a job.
Yeah, you got my job. I got too busy with life. So, and now I'm doing my master's at the moment at the University of Birmingham, and it's a master's in cancer research. So I just spend all of my days in lab.
Yeah. It's fun in the labs. They're hot.
Yeah.
We were in the labs today. We were at a research group meeting, and we were getting some tutorials and some molecular techniques and
way too warm.
Yeah, and we did 2 days worth of stuff and it didn't work.
Yeah.
It seems infuriating. I completely understand why our fellow colleagues are just angry all the time.
Well, we in Birmingham we have our way of keeping ourselves cool. So we have this big room, which is a cold room, which is basically a fridge, and it's 4 degrees. So when it's too hot and we can't deal, we just go there and sort the samples out or clean.
That sounds lovely.
Brilliant, yeah.
I could have done with that today.
Actually, there are 3 negative 80 degree fridges.
Well, that's probably a bit too cold.
But you can
I
just
don't fit in a fridge.
Or freezer,
yeah,
and you can just open it up,
whole room.
and then just leave the door open for like 5 seconds and then close it.
What about your samples?
Yeah, that's probably bad practice.
Yeah. Okay. All right.
Just ignore that. Sorry.
Okay, so let's move on to the topics of this week and what we've found in the news.
Yeah.
Now, I'm going to start this.
Okay, yours is interesting.
Because
I've read this. You go.
This is exciting. So
It's terrifying.
it is terrifying.
I promise it with that.
I like it. So I don't know how much you know about centipedes, but you'll probably find some in the garden, and they won't be much more than maybe 3 or 4 centimeters.
In the garden.
Normally really thin. You can get some thicker ones, but yes, they are very fast. That's what they're known for. They're predators, and therefore need to be able to move quickly to hunt other invertebrates. However, when you get to the warmer places, these centipedes get much bigger, like 20 to 30 centimeters.
Oh wow! Okay.
So
Like an inch thick, like an inch wide.
Yeah, between half an inch and an inch wide. And these guys are nasty. Like they have some quite potent venom in them and are really fast. They are quite popular amongst entomology hobbyists as pets.
The insane ones.
Yeah, one of our friends had one for a while.
Yeah.
Actually. But
I remember reading a story about how a guy got told that he asked his doctor about keeping Scolopendra, the big ones, and his doctor was like, "You have to come to me. Like, when you buy it, bring it to me, and we'll let it bite you. Because if you're allergic to it, you'll die. So bring it to me."
I've heard that as well. I've heard that.
If it bites you and you're allergic to it, we can fix that straight away. But then we'll know that you have to get rid of it.
Yeah.
Because if you find out when you're at home, you'll die. They're scary.
They're very much not handle pets.
No.
They're
30 centimeter tweezers if you need to move them.
Observing pets.
Well, I'm definitely more into dogs.
They bite you too.
Whereas these guys really excite me. But so one of the bigger genuses of these is Scolopendra. They're the big ones that everyone goes to. So there's some fantastic species. Scolopendra gigantea is a
Gigantea. Just thought I'd emphasize that.
is a common one that people bring up. I'm pretty sure that's the one that lives in caves.
Yeah.
That picks bats out of the air.
They hang off the ceiling and pick bats out the air as they go past.
Lovely.
They're fast.
Yes.
So the reason that genus has been in the news this week, this was actually an article posted last week to National Geographic, I think it was. Yes, so they found an aquatic living centipede. Oh. So this is it's just like any other centipede you'll find, but it was found in Thailand by an entomologist based at the Natural History Museum. And being a good entomologist, as we do, he was poking around a river looking for
Wasn't he on his honeymoon?
He was on his honeymoon. But if you end up in Thailand, honeymoon or not, you go
have to look
searching
under
for
rocks.
insects.
You have to dig up rocks.
Because that's what you do.
Look in bark.
So he dug up a rock and he found this centipede. But instead of scuttling towards the forest, it scuttled into the river and under a rock, which instantly rung a bell and went, that's weird. So the article just describes it with some difficulty. He then caught it. So, because they are quick, so catching it it's impressive. I'm glad he did. Because
would they be slower in water though?
I honestly don't know. I think it would be really interesting to watch them move in water because centipedes they have. So the difference between centipedes and millipedes, there are a few. But millipedes have two pairs of legs per segment, whereas centipedes only have one. And this allows them to go much faster.
They have different dietary requirements as well.
Yes, millipedes are herbivorous, and centipedes will kill the millipedes.
That's why I have a millipede, not a centipede, in my bedroom.
Yes. So with their one pair of legs per segment, as one leg moves forward, the other goes back ready to come forward. So as this alternates all the way up the body, they scuttle very quickly along. So it'd be very interesting to see if they still utilise that in water or not.
Because it would produce quite an eel-like movement.
Yes,
in the water.
I imagine that is how it moves around. So it's predicted that it eats smaller invertebrates. I think it might have a go at some smaller fish, knowing their hunting capabilities. If you've seen some of the, I know some of the on-land ones.
How would that work as like the fish?
I imagine it's similar to some eels, in which, as the centipede scuttled and hid under a rock, I imagine it could stay under a rock and wait for something to come past looking for cover.
Well, like an anchor to the rock.
Or perhaps not even an anchor to the rock, but just using it as cover or using a hole in the rock as cover.
What about using itself as bait? So you know how it's quite big, but fishermen hook like worms and maggots and stuff, and then plop that into the water. So if it anchors itself onto a rock and then just lets the current just like flap it about a bit, and then when a fish comes past, it can become rigid and take that.
Oh, it might. That's
fish
interesting.
That's inquiring to the free meal.
This guy is 20 centimeters long.
Yeah.
So he's about 8 inches long, which is a bit big and fat for a worm that you're looking at. Like the fish that it's looking on getting are probably quite little.
Yeah.
But the on land Scolopendra species have been. I've seen them get frogs, large geckos, bats. Like there's lots of documentation of these guys taking our stuff significantly bigger than themselves. Because they are ferocious hunters. If you're scared of spiders, you shouldn't be. Be scared of centipedes. They're much worse.
I'm scared of spiders. So yeah, thanks for that. I'm dreaming about spiders and centipedes.
This is in West. There's some videos on YouTube of Scolopendras just completely devouring the entire sort of fleshy, anything that's kind of wet matter on a rat. And just leaving
there's
the
a rat
behind.
one and there is a gecko one actually.
Yeah.
Both filmed by pet owners that own these. So it's quite interesting. You tend to feed them on similar stuff than like the large Scolopendra. You'd probably feed them the same stuff as you would feed a medium to small snake. So you give them like small mice and rats and things because these guys are a ferocious sort of appetite. Though I think similarly to snakes, once they've had a big meal, they don't need another for a while. But yeah, these guys strip flesh. They are fantastic hunters. Which is but this specimen was caught in 2001. So it's taken quite a lot of time to fully work out that it is a new species and that they have like found the correct thing and going over describing it and fully categorizing it. It's so I don't know.
Is
when
the behavior is the novel behavior not enough for it to be a new genus? So the morphology is what's keeping it in that genus, and it's the behavior that's making it a new species within the genus.
I would expect that they would go with genetics.
I suspect it was a genetic differentiation to work out that it was a new species, but the behavior is certainly so far unique. Nobody else had heard of any centipedes doing this. If a few years ago now in the news, you might recall that there was a diving spider discovered that preyed upon small fish. that lived. And is it called the diving bell spider? It creates it uses its web to create a small air pocket and uses it like an old diving bell, and therefore will swim. It holds the oxygen in, so it can swim up in it, breathe, and then swim back down, and therefore can live underwater for quite some time because it requires so little, and the silk is smooth enough that oxygen can diffuse out of the water into its air bubble, which is
that's amazing.
Which is fantastic. So land insects have been known to gain a niche back in the water, but it's the first we've heard of centipedes, and this centipede is a big invert. 20 centimeters is a big insect, and so having a hunter like that in there, I think, is fantastic.
Yeah.
Coming back to your question, how is that possible? How possible it is to this behaviour being just one individual, not the whole species? Obviously, now that it got confirmed, but
that is a good question, and honestly, I don't know, because with killer whales, this is quite the jump, but it will make sense.
Yeah.
With killer whales, different pods have different hunting strategies. So while they're all the same species, a pod in one place might eat seals by staying close to rocks, whereas a pod in another place might eat fish by beaching them. And so these behaviours are learned, and therefore it must have been one pioneering individual that then had the most offspring because of its new behaviour, giving it its edge, and then it teaches it to its children. And therefore these behaviours arise like that. So if we were talking about an animal like that, I think that could be more, I think that would be open to a possibility that this could just be an individual that's learned. However, with insects, they are much more primal in that a lot of their behaviour is a bit robotic, to be honest. So for those that don't know, I am on a master's degree in research at the moment, and I work in behaviour on insects. I'm currently looking at mate guarding in crickets. And the reason the behaviour studies work so well on insects is that they are really quite robotic, and you can get a lot of natural behaviours from them in the lab because they'll continue to do this through impulse, whereas a lot of so-called higher order animals will exhibit stress and unnatural behaviour and need other things to be taught to them by adults of the community that aren't as natural because they have a lot more thinking power themselves. So while I wouldn't say absolutely no, it's not just a behaviour that this individual has learned, I'd say it's unlikely. I'd say it's probably a species niche that it's gotten into.
It's terrifying to think you go to Thailand.
Yes, apparently that the tank that I have that I need to fill with a new insect, because my millipede unfortunately died, apparently I need to fill it with water to see if I can get my hands on one of these.
No, as your housemate, I veto that completely. So from the new home of an aquatic centipede, apparently, I'm still baffled by that, to the new home of Juno on Jupiter, which is a little bit further away than Thailand. So
tell us what Juno is, David.
Juno was the god of marriage and fertility in the Roman times.
I'm going to go with probably not that Juno that we're talking about, though.
No, but it was named after that Juno. That Juno was the goddess of gods, married to the god Jupiter, and this particular probe was sent to Jupiter.
Appropriate naming.
I got confused for a second. It's like, okay, we're back on track.
We've gone to an ancient religion podcast. Yeah, so that's the naming convention. It's named... this is an interesting thing. What is a backronym?
A backronym?
Yeah.
Is it an acronym from the back?
Yeah, you're not too far off. So the BBC called it a backronym, and I was like, what's a backronym? So I looked it up. But it's where you take the word that you want to use and then create the acronym from that. So you don't call it something long and complicated and then take the first letters and that becoms the acronym.
Oh, so you
start with the word and then from those letters you decide what the long complicated name is. So it's the Jupiter Near Polar Orbiter, and you take the JU, the N, and then the O from Jupiter Near Polar Orbiter to come up with the backronym Juno.
Oh, okay. I still think it's cheating, but I like it.
Yeah, I'm going to do it. Yeah. So it left Earth 5 years ago, and I have something really interesting to talk about the trajectory, but we'll get to that in a bit because I found it fascinating. It's not particularly to do with the article that was released and the news that's being released, so I'll leave that till the end. So Juno left Earth 5 years ago in 2011, and it reached Jupiter yesterday, I believe. It arrived on the 4th of July, I believe. It's very high and spooky, I believe. Its current orbit is a 53-day elliptical orbit, but it's going to be pushed in even closer to a 14-day orbit in mid-October. And that will mean its orbit will be only a few kilometers above the very top clouds of Jupiter.
Oh, wow.
So it's going to be super, super close, and no probe has ever been that close before. It's got 8 remote sensors and 1 camera. Because of the UV stress, the camera's not going to last particularly long, only for a few months.
Okay.
But the probe should be there for 2 years until February 2018. And it's going to sense the deep interior. It's going to try and confirm that it has a core, because there's a bit of a contention within cosmology, or whatever you call it, about whether there's a hard metallic core or whether the gases just persist all the way to the center and become sort of more and more dense, but still technically gas.
Oh, that will be interesting.
And another theory as to how Jupiter has developed its magnetic. Give me the word, Ian. Field. Thank you. Its magnetic field is through a liquid metallic hydrogen bit somewhere in Jupiter.
That's out of my depth and quite possibly out of yours. What's liquid metallic hydrogen?
I don't know. It was 3 words that went together in the BBC article.
That's amazing.
I have no idea, but maybe some of you that are listening.
If you know, can you tweet me and explain that to me? Because I don't understand liquid metallic hydrogen, if I'm honest. But so there's a theory that that's what's the bulk of the sort of interior of Jupiter. So they want to confirm or deny that as well. So they can look at the structure and chemistry to try and help better understand the planet's formation, which is about 4.5 billion years ago. And this in turn, because it's such a large body and it's sort of centrally, in inverted commas, located within the sort of diameter of the radius of the solar system, that it will really help to inform on the solar system formation theories as well that we have.
Okay.
So depending on how far out from the sun it formed, will help to plug a lot of the knowledge gaps of other planets and just the solar system formation in general. But this trajectory...
Okay,
yeah,
so
here we are, yeah.
So, interesting trajectory. Go.
Imagine you're looking down at the solar system. Imagine it's planar, and you're looking down at it from the top.
Yeah.
And the
I
sun...
got taller.
The sun is right in the centre.
Okay.
And then obviously you've got the orbits around the sun. So 12 o'clock's upwards from you.
Okay.
6 o'clock is towards your chest as you're looking. Well, I suppose if you're bent over looking down at it, and so forth. So Earth is the 3rd planet out. Mars is the 4th planet out. And when Juno was blasted off into space 5 years ago, it was done so then it would get just past the orbit of Mars, but it didn't have enough power to get any further. So it's sort of, because of the gravity of the sun, it came back in, attached in inverted commas again, to the gravity of Earth. So if you imagine the attach point on the clock that we've just established at about 8 o'clock, because the planet's orbit counts clockwise, so about 8 o'clock, and then it didn't release until about 3 o'clock.
Okay, so it's then attached for...
Yeah, and it's super increased its speed as well, because obviously it's going at a planetary speed.
Yes.
And then centrifugal force flung it out into the depths of space, past Mars, all the way to Jupiter. But if you imagine, so the first component is you have to make sure that after, so it took 2 years for its initial release from Earth and then back to Earth. So you have to make sure that Earth, when you release it and it blasts off from Earth, that Earth is going to be in the correct place 2 years later, or almost 2 years later, for it to then attach to the gravity of Earth at the correct moment, for it to then gain enough momentum and speed, so that when it reaches that 3 o'clock point and gets flung off towards 12 o'clock, that it has, all that enough energy in it to make that journey. But also, Jupiter has to be in the correct place, which, if I remember off the top of my head, was somewhere like 10 o'clock 5 years ago. So then Jupiter ends up at the 12 o'clock point 5 years later, because that's where we flung Juno.
That's fantastic.
Wow.
The planning behind that.
The logistics. Like I read about that like half an hour ago, and
Your mind got blown.
my mind got blown,
Yeah.
and I've been like flustering through it because I just found it so immense.
That's like next level of planning. And
Yeah,
stuff.
that is some serious organisational
Yeah.
I'm quite a planner, to be fair. I'm just like, I have 2-day list to make a 2-day list. And
Oh yeah,
to-do list.
yeah, sounds like
But that's just like next level.
That's
Wow.
some forward
That's
thinking.
impressive, yeah.
So
I'm going to take it way back to way more elementary than rocket science. But did you ever do that thing when you were younger, when doing like kick-ups with a football or playing with an American football or a rugby ball, where you'd try and throw it up in an arc so that you could run and then catch it on the other side?
Obviously.
So it's like doing that, but 2 years goes in between until you have to catch it again. Because we fired it off Earth, it went up just past Mars, came back down, and we managed to catch it in our orbit in order to throw it again.
yeah,
Like half a cycle later. The maths behind that is colossal.
yeah, that's insane.
That's fantastic.
I found that really, really interesting. It's not part of the main article, but that was the most interesting thing I read when I was doing my notes. So I thought I'd spend a bit of time on that. But yeah, so Juno is now around Jupiter, and it will be collecting a whole bunch of scientific data once it gets closer in mid-October, and I'll be looking really forward to seeing what comes out of that. And if you want to tweet anything about Juno or about Jupiter or the science of Jupiter, send across any news stories you have related to that, and we'll try and include those in the podcast in future. If we know people want to hear us talk about that, we'll absolutely do that. Yeah.
Huh. Well, that is amazing.
So let's crash land back on Earth, specifically Brazil.
Oh, yes. So I've read something interesting. So now more about serious topic that concerns everyone, and everyone got worried a bit, maybe too much. But so let's talk about Zika virus.
Yes, this is a big topic at the moment.
Yeah.
It is, yeah, especially with upcoming Rio Olympics.
Rio Olympics,
yeah.
I've seen it mentioned a lot in relation to
Yeah, I'm not sure if like everyone heard how many sports people pulled out of the competition.
Yeah.
Like our Rory McIlroy.
Rory McIlroy's pulled out. Yeah.
That's how you pronounce his name.
English isn't your first language. I'll let
It's
you off.
not. Sorry. And Rihanna just cancelled her. Is it a performance on a festival? I think.
I think so. Yeah.
Yeah. So basically, people are getting worried about this. Let's start with the basics. Zika virus. It's virus similar to dengue fever or yellow fever. It's spread by mosquitoes, but it can also be spread by sexual transmission. And the biggest worry is that, so we don't have any vaccines or medications, but so far because it's just only mild symptoms, and it's like a normal fever, and you can get unharmed. However, there is big issues because when you're pregnant and you're infected with the virus, it can spread to the fetus, and it can cause brain deformations, and particularly microcephaly, which is microcephaly. How do you pronounce that?
Cephaly? Yeah, that sounds.
Yeah, that was the right one. Basically, what it is, it's your brain doesn't develop fully; it's too small, and it obviously has its consequences.
Okay, yeah, so quite a serious issue then.
Quite a serious issue,
from
yeah.
what could be just your average fever to causing some quite debilitating problems.
You got there?
It originated in Uganda, actually, in Africa, but it's spread to America, mainly South America.
Okay.
So
do they know how that spread? Is it mosquitoes? Because I didn't think they lived that long in order to make that kind of... unless it was on boats. That makes more sense. If it was mosquitoes travelling over boats.
The primary host of the Zika virus is monkey. So it was spread by monkeys, and now actually humans got affected. But mostly it's the virus that's spread across the monkeys, and mosquitoes are biting the monkeys.
Yeah. Good old vectors.
Yeah. So the recent news relating Zika virus is actually the vaccines. So there was a accelerated Nature article published on 28th of June, so quite recent, about two potential vaccines for Zika virus,
Okay.
which is quite promising.
Yes.
So there was obviously, as it is with those kind of issues and human issues, there was a big collaboration between different universities, institutes, and in between different scientists. The main thing they've done was they tested 2 different vaccines, but only in mice so far. Both of them had a quite successful response, so it is quite promising. So the first vaccine was what it's called based on a genetic code. So basically they use a part of the virus, a protein, for example, that this virus has and produces, and they've used it as a vaccine. And the second one was basically they've used inactive, so like a dead Zika virus, so it's not going to cause any harm because it's not active.
Okay.
And all of the mice that've been vaccinated responded really well, so they didn't got an infection, which is great.
Yes.
And the control group that was exposed to the same environment with the virus got the Zika virus and got infected.
Okay.
So that was a big news. However, scientists, so there was one professor that from University of Nottingham that basically said, well, obviously we have to go and test that in humans, and as long as it looks promising, there are some serious danger because our immune system is way more complex than mice system.
Yes.
Although we are quite similar and my studies are quite reliable, it might be difficult.
Yes. If I'm not wrong, if I'm not wrong, one of the big appeals about mice is our brain chemistry in particular is particularly similar to theirs. Is that
there are serious like similarities, but with the immune system and
yes.
Well, there is you can go with mice. I know from the cancer research point of view,
yes.
There are quite big differences in cancer research. So if you, for example, mice have telomerase and they have longer telomeres, which is quite an important bit in cancer research when that makes a major difference between humans and mice, and that's how some of the drugs or some of the cancer cells respond differently. And there are some differences in the immune system similarly to human. And one of the worries is that these vaccines might be they can produce antibodies that are quite similar to the ones for dengue fever or yellow fever, and they can interact or contradict each other somehow.
Okay.
I haven't got into details, but in our human system, and therefore they can increase or accelerate the actual infection and reaction and make things worse for a Zika infection.
That's bad.
So so obviously there's still a lot to go through and to test. So
it normally takes quite a lot of time to get these things through the system as well.
So it's a serious problem now. The whole Zika it's now a pandemic, isn't it? I think it's been.
Yes, I think I read something that said that it had been
announced a pandemic. So as much as it's a serious problem and we want to prevent it as fast as possible, and so far it's only like a problem for pregnant women, and obviously they babies because the virus affects the fetal development. But we're trying to prevent that. So we're trying to accelerate the research, and we're trying to find obviously vaccines for that. And it's going to be a speed up process. There is still a long way to go. But obviously we reacting faster than like how we reacted with Ebola, I think, which and it's not as serious as Ebola infection as well.
Okay. So there's promise coming.
Well, and as well, the whole issue with people pulling out of the Rio or big stars cancelling the tours. As much as I understand the position, for me, it's a bit blown out of the proportion. So public might got scared that this is a serious virus coming out like as Ebola. But so far, it's if you're not pregnant and you're not at big risk.
Okay, so but what about being a carrier? If there's a lot of people coming in and out of a country,
that's an issue. So
then we could see a spread much further than it currently is.
It's been seen in the US already because, for example, I've got a friend that works in the US, in Washington particularly. He said they travel a lot to Brazil or to basically Latin America as well. because they did business together. So obviously that can cause a spread.
There's a link there.
Yeah. But it doesn't spread that easily. Although mosquitoes.
Yes.
You can't get rid of them, unfortunately.
No, it's,
I read a thing to do with CRISPR.
Oh, yeah.
Yes.
So that you can get rid of them.
Yes.
You can get rid of mosquitoes, but that's a different story.
Yes.
For another
That
day.
would be ideal. Can you imagine those summer nights without...
Well, you still have mosquitoes, they just don't carry malaria, or they don't carry...
Yeah.
For those that don't know, CRISPR is a new gene editing technique that is useful for research. Basically, is we can get a much more precise and clearer view of what we're manipulating and looking at, so we can be more precise with things like vaccines and resistance in bacteria and insects to really speed things along.
Yeah,
it's very up and coming in
cancer research particularly as well,
yes,
because you can basically get rid of some genes that are causing cancer, and you can then test. Yeah, it can be a therapy way, or you can just do research with that to investigate the. But that's probably a different podcast, isn't it?
Yeah, so we've gone for another half an hour about that. So there's prospects for Zika.
Yeah.
Well, not for Zika.
No.
For the prevention of Zika.
2 weeks ago, I've attended a really interesting conference, and it ended up with a talk, which is kind of off the topic, but there was a guy that is involved in which is called like lab on a computer on a USB stick. So when you can sequence the DNA.
Oh yeah.
And that's what they've been doing with Ebola and Zika as well. So it's very important to know the whole sequence of the virus, because you can then track where it originated and when different types of the virus split and how they mutate.
Of course, yes.
So it's very interesting from that point of view. And so they've sent a PhD student to Sierra Leone with this USB kind of portable sequencer.
Okay.
And it's live sequencing. So you just plug it to your computer and it just gives you a sequence live.
Whole genetic sequence.
Yeah, and you basically
that sounds like it takes a lot of processing
they've shown a picture, the photos of the PhD guy, and he basically put the whole lab. So usually sequencing for people that don't know, sequencing takes days, weeks,
a long time,
and you need to have like a proper lab with really, really expensive equipment.
Super expensive.
And it takes a lot of space as well.
It's not the same as CSI, where they get DNA and within 2 hours know who it is.
That's a lie.
It takes a long time to get a genetic profile.
So he basically packed a lab and that sequencing machine into the luggage suitcase. And he went to Sierra Leone and they've sequenced it in a hotel. But basically, they're doing that the same with Zika in a camper. So it's a lab in a camper. So they're doing like camper to sequencing.
Camper van science.
Yeah.
Camper van science. I really like that.
That is interesting. That is amazing. So there is a promise and there is a technology coming as well too.
Oh.
Yeah, it was very interesting.
Well, that's good.
It's been an excellent podcast. We learned about scary giant invertebrates that swim in water, probes that had to use Earth's gravity to
used as a
slingshot to Jupiter, and the Zika virus and how it's
work is being done.
Work is being done.
Yeah,
and
obviously.
Things looking up for it. So fantastic.
So we shouldn't be that scared.
Yes. In terms of the British Science Association, you can always keep up to date with what we have going on Facebook and Twitter, and of course our website, our wonderful website.
Links will be in the description for those, as well as all of the stories, like the origins for all of the stories that we talked about today.
Yes, and we have a branch meeting coming up next week. So we should be talking about the next events that are going to be put into motion. So
so summer and autumn events, and we're best to talk about those soon.
Yeah, social media is the place to keep up to date. Okay.
And if you have any stories that you see that you want us to look at or even talk about on the podcast, then send them across to our Twitter or to our Facebook. Share them within the community. We can get people talking about the different science stories of each week. That'd be fantastic.
Yes.
Oh yeah, or if you have any questions that we didn't answer in the podcast, tweet us as well.
Yeah.
Yeah, we can answer those in the next podcast or just straight away on the social media platform.
Yeah.
Yes. Excellent. Well, thank you very much for listening, and this has been your Intellectual Interlude.
Goodbye.
Bye.
Bye.